This conference will now be recorded.
Hello, Bence. And hello, everyone. I think we can start. And yeah. So I just wanna make sure that right now, do you see my whole screen properly?
Yeah, I think it works well.
Okay, and you can hear me well?
Yeah, I can at least. People can let us know if it doesn't work for some reason properly. At any rate, feel free to use the chat. We can see it. We should be able to see it, hopefully. And of course, feel free to also ask questions either in the chat or you can also use your microphones and interrupt us if you want to. It's fine.
Yeah, regarding questions, when I made the slides, I wasn't sure about how much detail and technicalities I should add. So if it gets too technical and you need to ask a question or you need me to move faster, just let me know. Or if it's too basic, again, just let me know. Don't hesitate. And I cannot see the chat, so Bence, if you could just stop me if there's any question.
Yeah.
Okay. So we can now start, I guess. And we are gonna present you neuroimaging methods tonight. It's me, Panagiota, and Bence, Farkas. And we are actually gonna present a very brief introduction to the field. I'm gonna present quite a few neuroimaging methods, and then Bence is gonna dive into more details regarding functional magnetic resonance imaging. So let's start. What are brain imaging techniques? And we can answer that question by saying that usually it's a non-invasive or minimally invasive technique that directly or indirectly measures the structure or the function of the nervous system in vivo. And we take advantage of many technologies and properties of the brain, like technologies like X-rays, radioactive labeling, and properties of the brain like magnetic fields induced by the brain's electric activity and electric fields, as well as paramagnetic properties of hemoglobin. And brain imaging, it's a field where are many contributions from other fields other than neuroscience, and where there are many applications outside the neuroscience field. And neuroimaging techniques are used from neuroscientists for research purposes, from physicists, chemists, and even physicians. And there are two main categories of neuroimaging. The first one is structural imaging, which is everything about anatomy and the different tissues of the brain. And the second category is functional neuroimaging, which indirectly or maybe even directly measures brain functions, neural activity. And apart from those two, basically tonight it's gonna be all about functional neuroimaging. And apart from those two categories, we can go further and group techniques according to how invasive they are, or if they measure neural activity directly, or if they measure something else that shows a neural activity, and whether there is a stimulation process, a process which alters the brain activity. And just for information, there are big projects right now going on collecting data from different brain imaging modalities, and basically the goal is to pull everything together and try to make sense, connect the pieces of the puzzle of how the brain works. And there is one first question about neuroimaging, and it's all about resolution. We have two types of resolution. It's temporal, which answers the question when something happened, and temporal resolution is how accurate we can answer that question. And we have the idea of spatial resolution, which it's basically how accurately can we answer the question of where did that happen. And as technology evolves, we want to achieve the greatest possible temporal and spatial resolution, and if possible to combine those two. And at the right side of the screen, you can see where different brain imaging techniques that we are going to go over tonight are regarding spatial and temporal resolution. And I just have to mention that we are talking about cognitive operations, which tend to be really fast, and we are talking about milliseconds or maybe max a few seconds. And we are talking about neurons and sometimes even components of neurons, which are really small entities. So we want to be as specific as possible. And there are two issues that we want to solve with the higher possible resolution. It's that there is high variability of brain functioning within individual and between individuals, and there's a lot of noise in brain recordings. And I hope everything was clear so far. And I'm moving to a second idea of correlation versus causation, which you have probably heard before, that correlation is not causation. And we cannot actually deduce a cause and effect relationship between two events because we have data, even high quality data, that associate the two. And most of the neuroimaging techniques we are going to see today are showing correlational data. So we cannot jump to conclusions about causal mechanisms or causal relationships between specific brain measures that we take, for example, from EEG, and the specific brain cognitive functions. And this causal role can only be demonstrated if we alter the brain function, and we do it in a controlled way. And this is only possible through neuroimaging techniques that actually stimulate the brain. And we are going to see, hopefully, some of those later tonight. And I wanted to start by saying that we started to establish those relationships between structure and function based on patients that had neurological disorders or had brain lesions. And those brain lesions can actually show us how they relate to a cognitive process. However, I'm gonna stress out that they do not show causation. For example, if I have a lesion at region A and I cannot perform an X task, it does not mean that region A necessarily is responsible for task X. I think it's X that I used before. It might be affecting some other region, which in turn affects the function X. And there is what we call single and double dissociations, and which is basically single dissociation is a lesion to the brain structure A disrupts function X but not function Y. So we can infer that X and Y are somewhat independent of of each other. And there is double dissociations, which are better than single dissociations, because single dissociations can sometimes be misleading. And what is double dissociation? It's when a brain structure A impairs function A but not Y, and a brain structure B, a lesion, in brain structure B impairs function Y but not X. So we can associate the specific structure with the specific function. And I'm gonna pass to our first section of neuroimaging techniques. I'm gonna start from non-invasive neurophysiology, which is basically measuring the brain activity directly. We are actually concerned about the electrical activity of the brain. And our first technique is electroencephalography or EEG, which measures the electrical potential produced by the brain over the skull and the scalp. So basically, if you have ever participated in an EEG study or you are going to, I'm gonna encourage you to do so. You just walk in, there's a cup that you put in. Usually you have a screen in front of you, but there are many different kinds of tasks that you might perform. And this cup has from 20 to some hundreds of electrodes on it. Usually we can add some gel or any other ingredient that can facilitate the conduction between the scalp and the electrode, especially if you have a lot of hair. And then those electrodes pick up the electrical activity of your brain. They go through an amplifier, and we can see the potentials picked up from each electrode. And those potentials are gonna be processed through algorithms, and hopefully we are gonna discover something. And the advantages of EEG is basically that it has a very high temporal resolution, and it's very practical to use, and frankly quite cheap compared to other types of brain imaging techniques. And due to its practicality and its high temporal resolution, it's widely used in brain-computer interfaces, and there are many startups at the moment which use EEG to, I don't know, for their purposes. For for example, gaming. But we are going to see more of that in the brain-computer interface lecture, which I think it's in 2 weeks. And yeah, but there's one downside to EEG, and it is low spatial resolution. Basically, because the electrical activity produced by the neurons has to pass through many different kinds of tissues, like the brain, the cerebrospinal fluid, the skull, the scalp. Each one of those has different conducting properties. So the current chooses to pass through the path of less resistance. So it's the location is being distorted along the way, meaning that what is going to be picked up by this electrode right here, it's not necessarily what was produced by the neuron right underneath it. And the localization of the source is an ill-posed problem because many different source combinations can result in the same reading in the scalp. And electroencephalography or EEG, there's a main field in the EEG research regarding brain waves, which are oscillating electrical voltages in the brain, which measure just a few millionths of a volt. And there are 5 widely recognized brain waves. They all have Greek letter names. And each of those brain waves is associated with a specific brain state. For example, an increase in alpha power is associated with reduced attention, and an increase in theta power is associated with engagement in a cognitively demanding task. And yeah, I should also note that the EEG signal, for example, this thing that you are going to record here, it cannot be entirely alpha activity or theta activity. It has many different components which can be identified through processing. And also the signal is composite. For example, even if we measure an alpha wave, overall, it does not necessarily mean that all neurons oscillate at the alpha frequencies. And another thing that it's not directly related to brain waves, it's that EEG signal patterns are quite consistent among individuals, although there are individual differences. And this consistency is used to indicate the state of the brain, and it is used as a diagnostic tool for physicians. For example, here in red, you can see that we can actually easily identify a seizure in an epileptic patient. And it's not only used in epilepsy, it can also be used in sleep research. It can also be used during anesthesia or during coma. And apart from brain waves, there is another field of research within the EEG research, which are the event-related potentials. And basically, it's what we use to examine how a specific task changes the brain's activity. Basically, we have the participant go over the same stimulus for many trials. We center the signal we get as in relation with the stimulus onset, and then we take the average, and we can get something like that. And specific ERPs, event-related potentials, are associated with different cognitive tasks. For example, the P300 that I added here, it's P because it's positive and 300 because it appears 300 milliseconds after the stimulus onset, and it's associated with decision making. And actually, it's a really powerful and really useful ERP that it's being used again in BCIs to show when a participant has made a decision. And is everything clear so far?
No questions have been so far.
Okay.
So
Okay. Just want to make sure that I'm still connected because I only see my
yeah.
slide.
Yeah, I think everything's fine.
Okay, great. So we pass to our second brain imaging technique for tonight, which is a technique that is similar to EEG, but this time instead of measuring electric fields, we are measuring magnetic fields caused by those electric activity. Basically, an electrical current produces a circular magnetic field that is perpendicular to the direction of current, and the MEG device measures this magnetic field, and it primarily gets measures activity which happens parallel to the skull, like here, for example, which is typically in the sulci of the brain. And basically what the hardware is, it's a helmet with sensors, and the input of the sensor from the sensors is linked to a squid device that can record magnetic field changes. The thing is that because the electric activity and magnetic field produced by the brain is so small, the MEG device should be in a room that is magnetically shielded from all external magnetic fields, including the Earth's magnetic field. And those squid sensors, which are actually very sensitive, must be kept cold. So it's not as practical as EEG, but and it's expensive, but it has the same temporal resolution and much better spatial resolution, because unlike the electric fields, magnetic fields do not get as distorted when they pass through different kinds of tissues. And we have the same idea with the ERPs, just this time we are talking about event-related fields, so ERFs. And I just added this table. We haven't talked about PET and Fmri yet, but I just wanted to show you that EEG and MEG have the same excellent temporal resolution. However, MEG is much better in terms of spatial resolution, but the cost of equipment is quite high. And I'm finishing non-invasive neurophysiology to pass to invasive neurophysiology. By invasive, I mean that there's an operation going on. And the first brain imaging technique is Electrocorticography, or ECOG, which is actually a grid of electrodes placed directly this time on the surface of the brain. So main idea is EEG, but this time it's not a cap you just wear. It's electrodes directly placed on the brain, and usually ECOG is used in patients that would anyway go through brain surgery. Usually epilepsy patients. And what's going on is basically that there's a first operation where the surgeon puts the ECOG grid on the brain. They need to do some measurements and localize which specific place causes the epileptic seizures in order to cure them. And basically, these measurements can take up to days and weeks before the actual operation about epilepsy. So in this meantime, the patient can volunteer to participate in research experiments. And basically, because the location the grid is going to be put on, it's determined by the operation. Based on this localization, you can only answer specific research questions. For example, you cannot answer a research question about audition if the grid is going to be placed on the motor cortex. And again, same idea as EEG, but this time much higher spatial resolution because the electrode is placed so much directly into the neuron, and the depth of the electrode varies. For example, you can have electrodes that are really deep in the cortex, and you can have electrodes that only slightly penetrate the cortex. And yeah, same data as EEG. And also another thing that I should mention is that electrodes can sometimes be stimulated and alter brain activity, which makes these very powerful. And then we have another neuroimaging technique, which is mostly used in animal models and very widely used in rats, which is called single cell recordings, and it's actually a measurement of the action potential produced by individual or sets of neurons in living animals. And most of the time, many times, not most, the animals can move and around the space quite without issues. And we are actually inserting an electrode through a surgery in the skull and into the cortex, and it can go really deep into brain structures until it's near the neurons we are interested in. And we can either let the neuron be right outside the neurons that we are interested in, or try to insert it inside the cell. And while if we insert it inside, we are gonna be picking up only the activity of one neuron, but we run the risk of damaging the neuron and it's gonna die. Or we can leave it right outside, but even if it's very close to a neuron, it's gonna be picking up activity from many neurons. So after the recordings, we should do a processing to determine which spikes belong to each neuron. And that's it about single cell recordings. It's widely used for memory purposes, and it's a powerful technique. And that's it about that. And I pass to non-invasive stimulation this time. We are now altering the brain activity. And the first neuroimaging technique that does that is transcranial magnetic stimulation, or TMS, which is actually a focal stimulation of the brain, which is, it's non-invasive. There's no operation going on. And the hardware, it's a tightly wrapped wire coil encased in an insulated sheath, which is connected to powerful electrical capacitors, which when triggered, can send a large electrical current through the coil and generate a magnetic field. This magnetic field can propagate inside the brain and alter the activity of the neurons. For example, a brief magnetic field, it's gonna cause the neurons to fire. Although we are not really sure about how this is done, we are not really sure, for example, if it causes the axons or the soma to depolarize. And basically, the TMS, not PMS, as I wrote here, sorry, can last longer than the stimulation and can inhibit or enhance neural activity. So the big advantage that we get from that is that we can disrupt the normal activity of the selected region and perform a task, and then perform the same task without disrupting the normal activity. So it's as if we cause a lesion that it's reversible. And even though I'm talking about lesions, TMS, if it's properly done, it's safe. However, again, the spatial resolution is not excellent. For this purpose, and we can only reach areas in the cortex, not deep in the brain. And for this purpose, there's research going on deep TMS. And yeah, it's a really powerful technique. But the results so far have been variable. There are a lot of variables going on. For example, we can stimulate at various intensities, different timings. We can have continuous stimulation, repetitive, or different frequencies. And then when we do an experiment, we should take into consideration both the timing of the stimulation and the timing of the task. So there's a lot going on, and yes, there's a lot of research going on, and it's very promising. Actually, personally, I know doctors that offer TMS as a cure of Alzheimer disease effects. And that's what I have on TMS. I'm not really sure if it's clear how does it work, but basically what you have to take from all of this is that we stimulate neurons using a magnetic field. And sometimes you can cause the participant to have involuntary movements of hands. For example, if you stimulate the motor cortex, and yeah, it's quite weird for the participants because they see their hand moving, but it's involuntary. But yes, most of the times there's no effect that you can observe, like hand moving. And another imaging technique that alters the brain's activity, it's transcranial direct current stimulation, which is actually the delivery of a constant low current to the brain via two electrodes. And basically, it's an anode and a cathode, as you can see here. And the neurons that are underneath the cathode are less likely to fire, and the neurons that are underneath the anode are more likely to fire. So, and again, the effects can last up to an hour. So we have the time window to perform experiments with the stimulation and without the stimulation. And repeated application of TDCS can prolong the duration of the benefit, if it's a benefit, from minutes to weeks. And usually the participants experience a tingling or an itchy feeling. And while it is a safe technique, the main risk that the researcher should take into consideration is not to cause burns on the skin of the participant. And very similar to transcranial direct current stimulation is the transcranial alternating current stimulation, which is basically the same idea, but this time the current is not constant like here in TDCS, it is alternating. It oscillates, and this rate of oscillation is another tool that we can use to modulate the brain function. Because as we saw earlier, different frequencies of brain oscillations, brain waves, are associated with different cognitive functions. So using TACS, we can induce those oscillations at specific frequencies, and we can also induce those oscillations at two different brain regions at the same time and see how this affects the performance in a task. And also there's a lot of research experiments that use both TACS and TMS at the same time. And with that, I'm finishing the brain stimulation part, and I'm going to invasive stimulation, which means that we stimulate the brain, but this time we do it invasively. Again, there's an operation going on. And one of those techniques is deep brain stimulation, which is usually done in patients with movement disorders, such as Parkinson's. And it involves an electrode which is implanted deep into the brain, hence we call it deep brain stimulation, and it can be turned on and off and modulate neuronal activity. Usually we implant it for motor disorders. We implant it in the subthalamic nucleus of the basal ganglia. And while it does have a dramatic improvement in those patients, I mean, because it has this dramatic improvement in Parkinson's patients, there's an intensive research going on about applying the same technique on other neurological diseases. For example, Alzheimer's or depression. And yeah, actually, it's a really interesting technique. But one thing that many times we don't take into consideration, it's the actual experience of the patient and how difficult is it to take a decision to have a brain operation that's actually going to be altering the way your brain works. And I invite you to look into that, because it's I don't know, it might be personal, but I really like this technique. So, and I find it really interesting. And yeah, so that's it. And let's move to optogenetics, which for now it's used only in animals, although the first human clinical trial has been recently approved. And it involves basically, there's a virus which is injected into the brain of the animal. And this virus targets specific cells or a specific brain region, and it constructs ion channels that are light sensitive. So basically, using light, we can either turn on or off the neurons having those ion channels. And again, there's intensive research. It's a relatively new field, but there's a lot of research going on that. And they are constantly developing new types of viruses that have different responses to different colors, different types of light, and the response can be very fast or quite slow, and the ions might be kept open for a long duration of time, or very transient for a short duration of time. So there are many research questions that can be answered with this technique. Also, you can see here that in this case, the rat is implanted with a light source, but we can also have experiments without implanted light sources. If we are going to use infrared light, which can pass through the tissues of the brain. And again, it's a very powerful technique because it's highly specific in terms of cells or brain location, and it has a really high temporal resolution. And yeah, that's all I have on optogenetics, and I hope it's clear. But if not, pose your questions. And I'm passing to my last section of neuroimaging techniques, which is indirect measures of neuronal activity. And by indirect, I mean that we are not directly measuring the activity of neurons, but we are actually measuring metabolic changes correlated with this neural activity. And I'm going to talk about positron emission tomography, and then Bence is going to take over and talk to you about functional magnetic resonance imaging. But not for now, PET. And what is PET? It uses a radioactive label compound, which the participant which we introduce in the body of the participant. And we can measure local variations in cerebral blood flow that correlate with mental activity. Basically, the more active neural area is, the higher the metabolic demand, the higher the tracer that is going to be picked up by the PET scanner. And it has a spatial resolution of approximately 5 cubic millimeters. I think it's even smaller right now. And a really powerful property of this technique is that we can develop radioactive label compounds for specific diseases. For example, right here you see a radioactive label compound which was designed for Alzheimer's disease, and it basically shows tau amyloid existence in the brain. Tau amyloid, it's a protein associated with Alzheimer's disease. So we can see that how increased tau amyloid levels are in an Alzheimer's patient, and we can see how it compares with a normal adult. And yes, that's PET. And I'm gonna stop right here. And sharing. And if you have any question, yep.
No, yeah, of course. If people have questions, then.
Yeah, if you have any questions regarding all you have seen so far, please go ahead. If not, I'm gonna give the stage to Bence.
I think we can have like a 5-minute break before I start.
Yeah, good idea.
You need to make me presenter first.
Yeah.
So in any case, we'll start in well, let's say 8. That would be best then. Let's make it nice and round. So it's an 8-minute break, I guess. So you can reconvene at 8, everyone. Okay. In the meantime, we'll set up so I can actually present this.
Yeah, I've made you a presenter, so try to share your screen now. Does it work? Okay, perfect.
Okay, cool. Okay. So, hey everyone. Can you hear me all right?
Yes.
All right. Okay, so we can kind of slowly start the second part of this lecture, I hope. As Panagiota said, this second part is going to be on functional magnetic resonance imaging, which is basically, in our classification, belongs to the indirect measures of neural activity. Before we do that, I wanted to add two things to our previous section. The first one is, I put this in the chat when we were talking about it, but I realized that obviously that won't show up in the recording. Is that when we mentioned that EEG, for example, can be used to detect seizures in epileptic patients, I thought it might be interesting to understand why that is actually useful, because at a first glance it seems pretty useless. I mean, everyone can see when someone has an epileptic seizure by just looking at the person. But a lesser known fact about epilepsy is that it can come in many forms, and it is only the so-called grand mal seizures which are associated with the convulsions, the loss of consciousness, and the loss of muscle control that people might be familiar with when they think about epilepsy. But there are also more localized seizures, which do not have these very obvious effects, which you would not really be able to see just by looking at the person. But with EEG, you can detect these as well. And these have some interesting local effects. For example, if people have epileptic seizures in the temporal lobe, somewhere here, they can, for example, have auditory hallucinations and stuff like that. So I thought that's an interesting tidbit. And the second thing that occurred to me is that when we talk about all these different techniques, the usual two dimensions that we consider, that we have seen, is of course that of spatial resolution and temporal resolution. So how small of an area, or how small of a, or how individual, whether we can tell individual neurons apart from each other, or how small of an area can be real localized, whatever neural activity we measure, which would be spatial resolution. And temporal resolution would be how fast of a time scale can we detect changes in. But another kind of aspect of measuring neural activity is scale, which can be also important. Because, for example, single cell recordings give us really good spatial resolution and really good temporal resolution. But what they don't give us is the ability to actually record multiple neurons, a large number of neurons at the same time, which can also be important, especially if we consider more systems neuroscience perspectives, which are all about how we need to have a more holistic view of large number of regions and a large number of neurons. And one recent, so a lot of the trends in methodological developments in neuroscience have been trying to find ways which give us all three of these things. A neuroscientist's dream is really good temporal resolution, good spatial resolution in a large number of neurons. And one recent example of this that I wanted to highlight is this preprint by researchers at the Rockefeller University in New York, who basically demonstrated that calcium imaging, which gives us really good spatial resolution and pretty good temporal resolution, is almost comparable to single neuron recordings. And they managed to record around 1 million neurons in the mouse cortex, which is pretty impressive. So one way in which the field seems to be moving forward is getting better and better at not only spatial resolution and temporal resolution, but just the scale of things that we can actually measure. So I think that's a really cool kind of state-of-the-art, or rather a really cool recent view on what state-of-the-art looks like when it comes to neuroscience methodology. So yeah. Now, let's actually do this. Okay. So now we're actually going to talk about the subject of my part of the lecture, fMRI. As always, feel free to ask questions. You can use your microphones as well, but I can also see the chat in this little window, so feel free to use that too if you want me to, explain anything better, or go slower, or go faster, or if there's anything that you don't understand. Right. So hopefully you should see my presentation as well as the little highlighter that I have.
Yes, that's great.
Cool. So as I've already mentioned, fMRI basically belongs to the non-invasive correlational neuroimaging methods. And the acronym stands for magnetic resonance imaging, with the optional F standing for functional. So what the acronym already tells us is that whatever we have has something to do with magnets and resonance and imaging. So the magnetic part refers to the magnetic properties of nuclei in atoms that we essentially make use of when we do fMRI. Resonance is the specific phenomena, the specific physical phenomena that we use in order to generate a measurable electromagnetic signal. We'll see how that works in a sec. Imaging refers to the fact that this signal that we originally measure in the frequency domain is then eventually converted into a spatial domain representation, so an image. And functional stands for the fact that there's basically two branches of MRI. The structural one, which we'll go through first, which only gives us information about how the, about anatomy, basically, so how the brain, or in fact any other organ, looks like. Whereas we can actually use this very same procedure to also gain some insight into function, so what neurons actually do. So in this part of the presentation, I will basically go through just some basic info about the machine, the Fmri machine, then a short historical look at the field and how it developed over the years. Then we'll go through the actual principles of operation of MRI, and I will hope to give you an idea on and some insight on what actually happens when people go into the scanner and how we can get from that to the nice, pretty images that we've all seen of brain areas lighting up in these pictures. Then we'll go through some example research questions, the answerings during the investigation of which I think has greatly benefited from MRI and neuroimaging generally. And then we go through some problems, which there are always some when it comes to science. And we'll end on some hopefully more hopeful sections about how the future of fMRI might look like. What are some recent developments in how we can go about analyzing fMRI data. And if we still have time, I also want to, or I also thought I should end on an example recent study that I think is a good kind of representation of what good current fMRI research looks like. So we'll end on a high note instead of ending on problems only. Right. So the Fmri machine, this is how it looks like. So this is a standard 3 Tesla magnetic field strength Fmri machine. It's a Siemens one. I chose this picture specifically because I think this is the type of machine that we have in Paris at the CENIA, which is the kind of the neuroimaging center of the ICM, so the Institute for Neuroscience and Nervous System Science in Paris. So one of these guys costs around $2 million, or about 1.7 million euros. So if in the unlikely case that I actually make it in academia and become a professor, if I wanted to have an Fmri machine just for fun at home, and to be able to make cool experiments without my annoying colleagues, I would only have to put away around 56 years of a full professor's salary to buy one. So there's still hope. Although I might find it pretty difficult to actually transport it to my home, given that one of these guys weighs around 13 tons. And the higher, better machines with higher field strength cost even more and weigh even more. So fMRI is not easy. And as a result, actually most organizations that actually have fMRI machines only have a handful. So you won't find dozens and dozens of these lining up, even in the most advanced neuroscience centers. I think the CENIA, for example, only has 2 MRI machines, and it's a pretty big research organization. So let's go through a brief history of the field. So firstly, the phenomenon that we'll go through in a sec of nuclear magnetic resonance was first demonstrated in the 30s by Isidore Rebai and colleagues in the USA, and then later on extended to liquids and solids. It was first demonstrated in gases, but it was demonstrated in liquids and solids a bit later by Bloch and Purcell shortly after the Second World War. Now, to actually use this for medical science and for generally image formation, you had to wait a bit longer. And it was in the 70s, starting with this seminal paper published in 1973 by Paul Lauterbur, a chemist, who first demonstrated or laid down the theoretical foundations of how we could actually use this phenomenon to form images. And later on in this same century, the people actually started doing that. The first full body scan, as far as we are aware, of a person was demonstrated by the American physician Raymond Damadian in 1977. This scan took around 4 hours. So it was, with the existing technology, pretty difficult to do so, but it was feasible, and that's the important point. And the first paper published on this was, I think, a year later by a different research group. Now, this was, of course, only structural MRI, and more generally for, not just neuroscience, but for medical science, generally speaking. fMRI, so specifically functional magnetic resonance imaging, the technique, the neuroimaging technique, was first demonstrated in 1990 by this seminal paper by Saiji Ogawa and colleagues working from, I think, New York University. Oh, no, actually, it was Bell Labs, an American research organization, who showed that we can actually use this very same technique to gain some insight into neural function. And after this, progress in this field has been very rapid as well. So shortly after a flurry of papers demonstrated the feasibility of this in various different domains. And since then, progress has been mostly with respect to getting better and better signals by more and more clever manipulations of how the magnetic field that we set up works, getting better and better statistical techniques to actually analyze the data that we get, and understanding the physiological basis of what we're actually measuring. Some key names in this quest have been that of Carl Friston, Marcus Reichel, and Nikos Logothetis. But this is just a sample of many, many researchers who have contributed to this. Just to get a glimpse of how popular this is, I just very simply plotted here the number of studies which have the keyword fMRI in the online database of PubMed. This is not the most sophisticated technique. For example, you can see that there are in fact some studies which have fMRI keywords before 1990, which is of course impossible because the technique didn't exist back then. So these are either errors or just acronyms for different techniques. But the point is pretty obvious that since the 90s, basically, there has been a very steady and consistent increase in the number of papers each year that are published with this technique. And this recent drop, of course, is not due to any decrease in the amount of interest in the field. It just means that the most recent papers in this field have not been added to PubMed yet, but people are still very much using this. In fact, the popularity of this technique is the reason why I thought that it might be worth spending a full kind of second half on getting a better idea on how this works. So how does it work? Well, what I will hope to give in this section is to give you an idea on how we can get from this. So getting someone to come to your lab and actually lie in this scary machine while we bombard their brains with magnetic fields and radio frequency energy, to the pretty images of brain areas lighting up that we see in the studies and in the newspapers. This section draws heavily, as in my presentation, on two sources, the senior training sessions and the EMIOS eMRI course, which you can access freely. And I highly suggest you do if you're interested in knowing a bit more about the basis of this, because this website has some interesting visuals as well, and some videos which may be more dynamic than my more static presentation. Of course, we'll have access to this presentation with all the links, the working links, so you can see for yourselves. So the first ingredient in our explanation of what is actually happening when we do fMRI and how we can get a glimpse of brain function with this starts from some, well, basically high school physics. So just as a reminder that the world is composed of molecules, which are composed of atoms, which are in turn composed of protons and neutrons. You can see the standard model, the Bohr model here. And what is important for us is that the nucleus of atoms, all atoms, So the particles that compose the nucleus have this particular property, which is called spin. Now, this is actually pretty complicated of what this means. So spin is a quantum property, which means that if we were to try to really understand the physical basis of this, we'd have to do quantum mechanics, which I don't think any of us are particularly well knowledgeable in. But for our non-physicist brains, we can kind of, in terms of classical physics, imagine what this property means is that you can imagine the protons, for example, which we care about, so the protons, spinning along their axis, which are represented here. So you can imagine what's happening is as if these little guys were literally, spinning around a particular direction. That is, of course, not what's truly happening, but it gives us a kind of analogy with which to work with. And the important part is that during this spinning motion, these little guys actually generate a magnetic field, and the direction that this magnetic field has depends on the orientation of the axis that these guys are spinning in. And every atom has this particular property, but when it comes to fMRI, we only really care about the hydrogen atom for various reasons. Basically, it has the strongest magnetic component too in the body, both because of the way its nucleus is set up, and because, of course, it is very, very abundant in the body because it's in water, and of course we are around 80% water, including the brain. So in fact, most of the signal that we measure comes from the spins of hydrogen atoms. Now, the second ingredient to what we need is that we'll need a way to actually measure this, because what I talked about is magnetization, and we need some kind of electric signal really that will need to be processed. And luckily, Faraday's law of induction gives us a way of actually measuring this. So there won't be many equations. In fact, I don't think there will be any more equations, but this simple equation is just Faraday's law of induction, which just means that whenever there's a change in a magnetic field, it induces a change in a corresponding electric field. So in this illustration here, we generate a magnetic field by passing energy or passing electricity in this little coil of wires here. And a magnetic field is generated, which has a particular direction, represented by these green lines here. And this induces a measurable electric signal in this other coil of wires here, in this hypothetical example. So this is really important for us because that means that whenever there's a magnetic field, if we have a proper setup, if we have a proper receiver coil made of, like, in the simplest case, for example, copper. Of course, we don't use copper in fMRI machines, but you get the point. It allows us to actually measure magnetization and kind of translate it into a oscillatory electric signal. And this oscillatory electric signal is in fact going to be the basis of our measurement of brain activity. So what we want then is to have a measurable component, a measurable magnetic signal, a measurable magnetic field. Unlucky for us, this doesn't usually happen in most cases because these little guys, the spins, which are in the hydrogen molecules of water, which are in our brains, usually have random orientations. So right now, the spins in my brain of the hydrogen atoms of my brain are oriented all over the place. They're not aligned with each other, which is a problem because, of course, we don't really have techniques to measure individual spins. We don't have the kind of nanotechnology yet to be able to measure a single spin's magnetic field. We only have to rely on a large number of these spins, the sum of their individual magnetic fields. The problem being that, of course, this sum, so this net magnetic field, is going to basically be zero if these guys are oriented randomly, because what we can kind of say is that these can be represented, the axes of these guys can be represented as vectors. And of course, if we sum up vectors, so kind of just points or lines with orientations, then if they are distributed randomly, then that sum is going to be zero. So there is in fact no measurable magnetization right now in my brain. So what we really want, first of all, as a kind of first step to our measurement of brain activity, is to get these guys to somehow align so that there will be a component that we can measure. Luckily, we can't do that. And the way we do that is magnets. So we induce a strong magnetic field, a strong external magnetic field, which, due to something that is called polarization, so this effect is called polarization, will basically cause these spins, so these little guys, to align in particular directions. Specifically, if we imagine the magnetic field as this line, as I represented here, as a vector, because this has an orientation as well, it's standard to call this B0, but it doesn't really matter. This will basically cause the spins to orient themselves in one of two states. Either both of these states are parallel to the orientation of this external magnetic field that we produce, but some of these spins are going to be in a high state, which means that they will be pointing towards in the direction of the magnetic field, and some other guys are going to be anti-oriented, so they're going to be parallel still, but in the exact opposite direction. Now, if there were an equal number of these guys, then we wouldn't be very much, further, because of course our net magnetization would still be 0, because there will be an equal number of guys pointing in this direction and in this direction, which means that they would just cancel each other out again. Luckily, though, for various reasons that we won't go through, there will be a slightly larger number of little guys which are oriented in the high state, which, if we represent this in a coordinate system, and if we represent the net magnetization, so the direction and the strength of this magnetic field as a vector in this three-dimensional Cartesian space, then this means that we'll have a measurable magnetic component that is parallel to this external magnetic field and points towards this external magnetic field. So I chose to represent this magnetic field as a vector as well, but in fact I could have probably colored the entire background this color to give you the idea that really this all takes place in one giant, massively strong magnetic field that we generate, which has a direction. So I just chose to emphasize the direction at unit because it's important. But in fact, this is all within that field. It's not, it's not in one particular place. It's everywhere. So this is what we do, in fact, when we do fMRI. And just to give you an idea of how strong this magnetic field actually has to be, the Earth's general just geomagnetic field that all of us are exposed to right now at this moment is around 0.000005 Teslas. A magnet that we have on our refrigerators probably operates on a scale of around 0.001 Teslas. So standard clinical MRI, which is non-functional, operates around 1.5 Teslas, so around a 30,000 increase in strength. And fMRI, so functional MRI machines, tend to work in even higher magnetic field strengths. Generally speaking, the higher your magnetic field, the better your image is going to be. So nowadays, the best MRI machines tend to be around 7 Teslas. Now, this obviously causes some problems, and it can be pretty damn dangerous if you're not careful. So you really don't want to step in an fMRI machine with a wristwatch on, or if you have a pacemaker, for example. So people who have any kind of a metallic object in them for medical reasons, or for any reason, then they cannot take part in MRI experiments. Because if they did, things like this would happen. So here a steel shackle is thrown into a 4 Tesla magnetic scanner, and it gets stuck. Now here a stapler gets thrown into it, which gets immediately disintegrated. And obviously you don't want to be near that when that happens. Right. So now we are in a position where we have a magnetic component that we, in theory, should be able to measure. In practice, though, we're still not quite there yet, because actually measuring this little magnetic vector is still problematic. The reason for that is that this is, as I've said, parallel to this magnetic field, this giant magnetic field. So in practice, this is going to be really small with respect to the magnetic field, which means that we actually cannot measure this if it stays in the direction of the giant magnetic field, which means that we have to somehow perturb the system so that it actually moves directions somehow. Because really, we really want the best of both worlds. We want the spins to be kind of oriented in a particular direction so that they actually give a signal to measure, but you also want this signal to not be in the direction of the magnetic field, which causes us to not be able to measure it. And the way we do that is we make use of resonance, finally. Basically, what we do is that we apply a radio frequency, a pulse of radio frequency energy, so just electromagnetic energy to the system, to the brain. We send a radio frequency pulse to it, which has a particular kind of predictable effect on the system. What we say is that it causes the spins to flip, which just means that they change orientation. And the way they change orientation depends on how we set up this radio frequency pulse and which direction this pulse comes from. So if we set it up correctly, and if we're clever about it, we can get the magnetic field, so the net magnetization that the spins generate, to be exactly perpendicular to the massive magnetic field that we generated. And that means that if we now have a measure, so a receiver coil, remember Faraday's law of induction, that is located perpendicular to the massive magnetic field, then we will finally be in a position where we can actually measure this component. And what we measure is really relaxation. So what we do is that we first of all set up the polarization. So we turn on the massive magnetic field and orient the spins. Then we excite these spins so that they give a signal in the perpendicular direction. After which the spins, we turn off the RF pulse. So the spins are going to be now in a phase called relaxation, where they will slowly recover to their original orientation. And what we can measure is typically a signal that looks like this. This is known as free induction decay, or FID, and this has a particular type of temporal property. So it's an oscillatory signal that is highest. So imagine that we applied the RF pulse at time 0 in this figure. So shortly after which our signal that is measured in this orientation, so perpendicular to the massive magnetic field, is going to be maximal. And as the signal or as the spins recover through relaxation to their original orientation, the signal in this direction is going to be maximal again. But remember, we can't measure that. And however, in this direction is going to then decay back to 0. And what we measure then essentially is this decay of the signal back to 0 slowly as the system recovers. Well, by slowly, I mean slowly in terms of physics, which still means milliseconds or microsecond timescales. And crucially, what actually allows us to, or what makes us care about any of this, is the fact that this relaxation, so the time of the relaxation, here I only plotted the envelope of this signal, so how fast this decays, but in fact it looks oscillatory still, but I chose to ignore that in these figures, is that these relaxation times depend on the type of tissue that the signal came from. And particular type of tissues will have faster relaxation times than others. Which means then that if we actually measure this, then we can kind of make a guess, or well, not really a guess, we can in fact know for sure which type of tissue this signal came from. And then we are in a position where we can actually finally convert this back to an image. So there's still some remaining magic here. Obviously, there's still a long ways to go from this type of signal to this image. And I won't go through how that works, but you can just believe me that it does work. There's some clever use of sequences of perturbations to the magnetic field, along with some fancy maths to actually translate this. For those of you who might be more math savvy, the idea of transmitting or translating temporal information or information in a temporal domain to the spatial domain might ring a bell, as we probably use Fourier transforms to do this. And in fact, we do. We use a sequence of Fourier transforms. But the point is that it does work. So we're almost at the pretty pictures yet. We're at the pretty structural pictures, but I haven't actually told you how this relates to brain activity at all. Now here comes the second step, which is actually going to be a lot less technical. So what we do is that really what we're measuring with fMRI is not actually tissues directly, and it's not actually neurons at all. Rather, it is blood. The reason for this is that blood also has magnetic properties, specifically hemoglobin, so the part of red blood cells, which actually has oxygen attached to it, and that actually carries oxygen in our in our cardiovascular system. Also has a particular type of magnetic, it has particular magnetic properties. Specifically, it is diamagnetic, which means that it increases magnetic fields when it's oxygenated. So it will increase the overall fMRI signal or MRI signal that we get. And it is paramagnetic, which means that it kind of decreases, it distorts magnetic fields when it is deoxygenated, so when it doesn't have oxygen attached to it, which means that blood that is rich in oxygen is generally going to increase the signal that we get, and blood that is oxygen-less, relatively, is going to decrease the signal that we get. And neural activity, of course, is extremely energetically costly, and it needs a lot of oxygen. So much so that, in fact, for an adult, a human, around 20 to 25% of the resting metabolic rate, so the overall number of energy, basically quantity of energy that you use during rest, is devoted to the brain, even though it only takes up a couple of percent of body mass. The brain is surprisingly light. It's just barely above a kilogram. So it needs a lot of oxygen. So at a first glance, you might think then that, okay, so what we see then is that when there's a lot of neural activity, then there's going to be an overall decrease in the MRI signal, right? Because then the neurons are going to suck up the oxygen, leaving relatively more deoxygenated blood, causing a decrease in the signal. But it's actually a bit more complicated than that. And specifically, the response that we get tends to be an increase in the signal. The reason for that is due to a phenomenon known as neurovascular coupling, which I won't get into because it's actually pretty complicated and not very well understood, surprisingly. There's still a lot of unknowns about how exactly it works. But what the overall picture is that what tends to happen is that there is at first a small dip that corresponds exactly to what I've said, our intuitive notion that, okay, the neurons suck up the blood or suck up the oxygen from the blood, leading to a decrease in the electromagnetic signal. But the vascular system actually responds to that. It detects through receptors that there has been a loss of oxygen, and it responds to that by massively increasing blood flow to regions that have had a higher metabolic activity. And this increase tends to be a massive overshoot of the actual metabolic demands of the neurons in that area. So in a sense, it causes much more blood and much more oxygen to flow into that particular area. The neurons actually have the capacity to suck up. So this will overall then cause an increase in the MRI signal. And this is very, very consistent, so much so that it has names. So therefore, the signal specifically that we measure in fMRI is called the blood oxygenation level dependent, or BOLT signal. And it tends to be represented by a percentage change in the electromagnetic signal relative to some baseline. And this has a particular characteristic shape known as the hemodynamic response function, or HRF. And this is what it looks like. It tends to be very consistent across regions. So if, for example, a neural activity actually responds to the stimulus that we present, then you'll see that first there will be this initial dip in the BOLT signal, then this very sharply peaked increase, and then a slight overshoot, and then it will return to baseline. And as you can see, this happens in a time scale of seconds. So the way we actually analyze this is fairly straightforward. We basically do regression, known as the general linear model. So what we do is that we design a particular task that we think will engage a cognitive operation that we're interested in. And then we kind of make a prediction of what the neural activity that actually responds to this task should look like by convolving the hemodynamic response function with our variable. And this gives us kind of prediction of what a area of the brain, so what a voxel, so particular kind of located. A voxel is basically a pixel, but it's three-dimensional, and it tends to be the basic unit that we kind of partition the brain into when we talk about fMRI. So for each voxel, we generate this kind of model response, and then we compare this to the actual response that we observe, and then kind of make a statistical test comparing how well this fits. You can see this in this little video that I think I'll show it to you. So here, imagine that these are two voxels, and the task here is, for example, we care about localizing the part of the brain that actually does motor control. So the part of the brain that is responsible for example, this simple finger movement, right? And what will be plotted here are the actually measured bold time courses for two example voxels. One is actually located in the motor cortex here in red, and one in the occipital cortex in yellow. And the motor cortex, of course, will be responsible for generating this movement. So the way it will look like is going to be very similar to our task, whereas the occipital cortex won't really care about this movement, so it won't look anything like it. So we acquire the data, and there's going to be some pre-processing steps that I won't go through, but basically we kind of align each participant's brain into a standard template, and smooth the signal so that it looks better. Then we generate this time course by, as I said, convolving this with the hemodynamic response function, and then we compare the measurements we get from the two time courses, from the two voxels. And what we'll see is that, of course, this corresponds to our variable much better than parts of the occipital cortex. And we kind of quantify this using a statistical test. And basically, what gets actually plotted in these images is the statistical test's number. The higher it is, the better the correspondence. And what we basically usually do is that we threshold this. So we only plot the parts of the brain that are sort of voxels where this statistical test has a value that is higher than a particular threshold that we choose. Let's say that the threshold is 3, and that this voxel here only has a value of 1, whereas this 5. So this will get plotted, but this won't. And that is actually how we get finally to the pretty images that we see in the journals. And we can basically have two kinds of experimental designs. In a block design, which we've seen, our stimuli will be presented in a short sequence. So there will be a kind of block-like activity for like a couple of, let's say like half a minute. But we can also do event-related designs, which are much closer to what we do with like EEG, for example. But you'll see that these events still have to be relatively far apart, and the time scale is still very much seconds. But we can do event-related designs in fMRI too, but they'll have slightly different timescales and slightly less power to detect big or to detect small effects. So we're finally here. So hopefully you now have a better understanding of how these images are generated. So really the key take-home message here is that I want you to understand that lighting up in these images doesn't mean some kind of ground truth. So we don't really directly observe the neurons lighting up or doing anything. What we observe is a relative increase in oxygenated blood as a result of increased neural activity, which is detected through a pretty complex pipeline of statistical analysis and using magnetic wave fields and radio frequency waves. Right, so now we've understood, hopefully, how fMRI actually looks like and what it does and how it works. But I haven't actually really tied this particularly precisely to neural activity yet. I've been deliberately kind of vague on what kind of neural activity this actually means, because we're pretty far apart from individual neurons transmitting action potentials. I mean, what we talk about is the really networks of neurons. Just as an illustration, a typical voxel, so the smallest unit that we can actually detect with fMRI, contains about 5.5 million neurons, and around 2.2 to 5.5 to the power of 2 times 10 to the power of 10. So around average 30 billion. This is, I think, synapses, and 22 kilometers of dendrites, and 220 kilometers of axons. So we're pretty, we're working on a pretty large timescale here. So really, what we're measuring here is the collective activity of excitation inhibition networks of neurons. So this paper by Logothetis, again, you'll have the link to this, is very good. It explains quite well what the kind of signal that we're measuring is. And basically, the key idea is that there is no simple one-to-one correspondence between excitation and so action potentials and excitatory cells and bolt activity. So we can, in fact, for example, get increased bolt signals even when it's inhibitory neurons, represented by these blue lines here, increase their activity. So the correspondence is a bit tricky. The general consensus is that what we're actually, when we see a neural region with a increased bolt response, that generally represents inputs to that area and the local processing of that input. So really, when we observe an increased bolt signal in a particular brain region, what we're really measuring is the action potentials arriving at that location, which originally came from other parts of the brain. And we're not really measuring the action potentials that are generated and travel from that location. This might seem unimportant, but actually it is quite crucial in how we interpret the bolt signal. And for example, if you also follow the reinforcement learning lecture that I'm going to give in a couple of weeks, we'll see that this has some important implications when we interpret the kinds of fMRI research that has been done in that area. So in the next sections, I would like to basically give you an idea on what fMRI has actually given us. And hopefully, much like the Romans in this famous part from the life of Brian, quite a lot I hope to give you an idea about. But just to give you some clues that this question of whether neuroimaging is actually useful is not just like some theoretical stuff. People do actually question this and have been somewhat skeptical on whether this has been actually particularly conducive to cognitive neuroscience. For example, the neuropsychologist Max Coltheart has this relatively famous article where he's somewhat skeptical about the use of fMRI in cognitive neuropsychological research. And this is quite fair, given that a lot of early work using fMRI has mostly confirmed already existing theories. And by the very nature, people were initially interested in kind of just giving people an idea that demonstrating that MRI actually does work in detecting functions. So a lot of early work did concentrate on confirming existing theories. For example, a lot of work has been done in investigating visual system, like this study by McCuffrey and Zeki has basically confirmed previous theories and previous data that came from monkey electrophysiology and neuropsychological, so study of brain damaged people, which highlighted the specific area now known as V4 in the occipital cortex as being the color center, so the part of the brain that mostly processes color information. But I would argue that there have been areas where neuroimaging has been much, much more crucial and has really contributed a lot to our understanding of it. And I think one good example of this is face processing. So the general idea that faces are really quite important for primates, including humans, is, it's not particularly surprising. But whether the human visual system and the human cognitive system has a specific module for processing faces is a lot more of a novel idea. And by finding a brain region that does seem to be pretty selective for processing faces has been quite crucial in promoting the idea and the hypothesis that there does seem to be, in some sense, a dedicated module to processing face and face-related information. So this area, known as the fusiform face area, or FFA, is located in the bottom of the brain. So this is the view from the brain from below, and this is the back of the brain. This is the front, and the FFA is located in the back here, still in the junction of the occipital and the temporal lobes in the bottom here, in both sides. This first has been demonstrated using PET imaging actually by Sargent and colleagues, and then really investigated a lot and given the name FFA by the neuroscientist Nancy Kanwisha, who I think is at MIT, in this very influential paper in 1997. So a lot of research on this area has been basically about finding out why exactly the FFA exists. So there are two extremes of this kind of view. Either the FFA is due to genetic, hardwiring, so it develops in everyone due to the fact that the brain due to how the brain is wired up, or it responds only and develops only as people acquire expertise in processing phases, and so as a result of environmental input. And there is some evidence for both, and both comes from fMRI research. So evidence for the idea that the FFA has something to do with the input, so the environmental input that people actually receive, comes from this study, for example, by Gauthier and colleagues, who wondered whether they could get the FFA to also respond to other stimuli if people are also experts in identifying differences in those stimuli. Because really what is special about faces is that they look very similar, and differences between them, between individual faces, are very small, but humans are very, very good at, detecting these differences. So they wondered whether people who are just as expert in detecting subtle differences in other visual domains might also activate the FFA. And they showed that this does indeed happen. So people who are experts in cars, for example, actually activate their FFA to a significant degree, much more than for birds. Whereas people who are experts in birds, so ornithologists, amateur or professional ornithologists, activate their FFA in response to trying to detect subtle differences in between different birds, but not for different cars. So clearly, environment does have something to do with this. But there's also some pretty solid evidence that genes are in fact involved, and that there is something special about the FFA that is kind of independent of visual input. And one recent study has been by actually Nancy Combisher and colleagues, published in 2020, which has shown that in fact the FFA also responds in much the same way in blind people. So in people who are blind from birth. What they've shown is that when these people are given faces as stimuli, but they use haptic information, so they're very well developed touch sense, and they kind of perceive faces in their usual way through touching them instead of through their eyes. They also activate the FFA, much the same way that people who are sighted visuals do. So clearly the FFA is kind of a mix of both. And I don't think I have to stress just how important neuroimaging has been in actually investigating this. Now, another area and another network, in fact, that has really benefited from neuroimaging, and in fact, this entire research area really only exists due to neuroimaging, is the default mode network, or the DMN. So what this is that during the early years, so the 90s basically, people have done, as I've shown you, a lot of neuroimaging research, and they have been doing a lot of different tasks. And a couple of researchers, for example, Markus Reichler and colleagues, have noticed that people mostly focused on the increases in neural activity between different conditions, between a baseline condition, which is usually like a resting condition where people did not really do anything. They just lay in the scanner and just did nothing, versus task. So this contrast of activity, researchers really focused on the increases, but there were some pretty interesting patterns in what brain areas decreased their activity during the execution of tasks compared to rests. And what they have noticed is that these decreases tend to be pretty consistent. So in fact, no matter what tasks people were engaged in, they seem to deactivate the same set of areas. Specifically, this area here, known as the posterior cingulate cortex, the PCC, and this area in the front here, known as the medial frontal cortex. And what they've shown is that these areas are also the ones that are most metabolically active during the resting conditions. And they deemed this network of brain regions hypothetically the default mode of brain function, or the default mode network. And the general idea is, and there's has been a lot of research in this, is that basically this brain network is active whenever you don't do anything, but it still has a function. Specifically, what people think this brain network is involved in is spontaneous thought and mind wandering, and the kinds of mental activity when you kind of turn your attention to stimuli that are inside you, so inwards, so kind of memories or imagining hypothetical scenarios, things like that, that your brain kind of constructs by itself, from itself, instead of the actual environment around you. Obviously, this research has been very closely tied to neuroimaging. And this is from PET, but of course, it has been investigated in fMRI as well. And more generally, I think a crucial new perspective that fMRI has given us is the idea that really what we should be looking at is not just individual brain regions, but rather networks of brain regions. And one way in which you can also use fMRI data that we said we don't really have time to get into is to look at connectivity. So if you just put people and have them rest inside the fMRI scanner and then look at the time series of the bolt signal, what you can do is correlate the time series at different parts of the brain to each other and kind of estimate that, you know, if two time series have a high correlation, then that probably means that those two brain areas probably have either a direct or at least an indirect connection with each other. And what you can do is then kind of group brain areas based on what other brain areas they have high correlations for. And what pops out is that there really seems to be a large-scale network organization that is inherent to the brain, to the cerebral cortex at least. So if you kind of look at these big connectivity patterns, one very robustly replicated pattern of connectivity tends to be this so-called 7-network solution. So that at rest, these brain regions tend to be, that are color-coded by the same, you know, colors, tend to be very highly co-activated. So their neural activity, the activity that they produce tends to be very highly correlated within their respective networks, but not necessarily across the networks. And these 7 networks specifically tend to be, you know, very universal. And this gives us kind of a different perspective on the whole thing, on the whole field, because rather than localizing brain function to an individual area, what you really have to consider is the kind of broader network that that area belongs to. Of course, it still means that different areas do different things, you know, as has been very conclusively demonstrated by neuropsychological experiments and causal manipulations. You know, if you knock out the occipital cortex, people will still have visual impairments. But rather than only being localizable to this specific area, what you have to consider is how this affects the whole network. Now, I briefly want to talk about some common issues that pop up in fMRI and also in neuroimaging more generally. One of these issues is the problem of multiple comparisons. So in this pretty hilarious paper, in fact, it's first a conference poster in the Human Brain Mapping Conference of 2009, and it was then made into a kind of fake paper. But what they did is very real, is by Craig Bennett and colleagues. And what they have done is that they put a fish, a dead fish, a salmon, inside an fMRI scanner, and they had it basically undergo a perspective taking task where they showed it images of humans in various situations, which when presented to human participants tend to kind of induce perspective taking and empathy. And what they've shown is that using the standard way of analyzing fMRI data, they've shown that, well, the fish seemed to have been engaged in perspective taking, in that it had a couple of pretty significantly activated voxels in his brain. So either there's a massive scientific discovery or there's something kind of going wrong here. And of course, sadly, it's the latter. So the problem here is that, briefly put, what I've said is that, remember when we talked about how we actually analyze fMRI data? What we do is that basically we make a particular statistical test for each voxel of the brain. And the problem is that if we use the standard way of determining whether a statistical test is significant or not, which is of course the p-value, so the value of statistical significance, then that means that if we do this procedure a large number of times, remember there's around 60,000 voxels in the brain. So if we just repeat the procedure a large amount of times, then we'll eventually going to find something, even if there is actually no effect there. So this, of course, has a solid statistical foundation, which we will not get into. But basically, you need to control for multiple comparisons. And whenever in your readings you come across a paper that shows these kinds of maps, but it says uncorrected, or it doesn't explicitly mention that these statistical maps have been corrected for multiple comparisons, you should be very, very skeptical. The second problem that is also common to different neuroimaging techniques is called the reverse inference. So imagine that I give you this reasoning. In the present study, when task comparison A was presented, brain area Z was active. In other studies, when cognitive process X was engaged, then brain area Z was active. Thus, the activity of brain area Z in this study demonstrates the engagement of cognitive process X. To give you maybe a more concrete example, let's imagine that I did let's say like a visual task where I just asked people to identify like different faces. Let's stick to faces. And I observed that, well, actually there has been activity in the right inferior frontal gyrus. This is the left one, but I couldn't find a nice picture of the right one. And I want to explain why that was. I might then turn to this pretty influential paper by Aaron and Poldrack, actually, which demonstrated that the right inferior frontal gyrus is actually involved in behavioral inhibition and the inhibition of prepotent responses. So I might then say that, well, the fact that this area was activated means that there's probably a component here that was activated by my experimental task that made people inhibit prepotent behavioral responses. And this might seem reasonable, but of course the problem here is that we're not at the point where we can really reliably tell which brain area is associated with which exact cognitive function. And if I do a bit of digging, which I've done, I can also find that the right inferior frontal gyrus is activated in when people lie and tell untruths, when people mentally rotate objects, when people feel pain, when people engage in interoception, and a whole bunch of other things as well. So really, I can't tell which specific aspect of this is the most important. I can't really tie any of this specifically to any specific cognitive function. So you have to be careful when you make this kind of inference. Generally speaking, the graphically represented here, the strength of the reverse inference process is going to be dependent on which other cognitive processes also engage that specific observable fMRI activation pattern. If it is very selective and there really is only one cognitive process that actually engages this, then the logic is going to be sound. But if there's a whole other set of cognitive processes that also has links to this specific brain area, then reverse inference won't be valid. So a couple of ways to not do reverse inference or to mitigate the problem. But the first, of course, is to not do reverse inference. So to just not actually feel the need to specify or to hypothesize about what brain areas that you did not expect to find might be doing in your task, because you don't have a specific hypothesis for them, so just ignore them. Maybe some better solutions are to find converging evidence for the involvement of the cognitive process that you're interested in. So for example, if in my task, okay, I can find some nice behavioral signature of inhibition. For example, if I can demonstrate that people in certain cases were much slower, which tends to be a reliable signature that they were engaging in inhibition because they had to inhibit their prepotent response, and as a result, their reaction times would be, slower. Then I might be a bit more confident in interpreting brain region activity as belonging to a particular cognitive function. Also, if I can incorporate prior information about the specificity of the region, then I can also inform my interpretation. So for example, if the region that I'm talking about is very small and really based on the previous literature only associated to a small number of cognitive functions, then reverse inference will be much more valid than in the case when I talk about a really broad activation in a large number of areas, which are each involved in different types of functions based on the previous literature. And the final problem that I wanted to talk about is related to the analytical flexibility of fMRI. So I already emphasized that the way we get to the pretty images is through a pretty complex pipeline of statistics and individual choices that researchers have to make. And this can be problematic. So in this study by Botvinnik, Nezer and colleagues, what they've done is that they basically given the same data set, the exact same collected data set to 70 teams of fMRI researchers, as in like 70 different research groups all around the world, which kind of specialize in fMRI research, and ask them to verify to confirm or reject the exact same nine hypotheses about this data set. And what they try to estimate is how consistent these researchers are in both their final answers and in the analytic pipeline that they used. And the results were kind of worrying. So in fact, no two teams, not two teams, have chosen the same statistical technique. So obviously there are more subtle differences than, like no one really did anything crazy. But the fact that you couldn't find two fMRI teams that have actually chosen the exact same procedure can be kind of worrying. And even though the correlations of the threshold statistical maps, so the actual pretty pictures, was relatively good, at least above 0.5, the correlations in the unthresholded maps that those thresholds are based on was much, much lower. So, this can be problematic. So there are some solutions to this as well. First of all, if you share the analysis code, then it will allow people to actually, well, reproduce the results and see how the choices that you made might affect them. You can also perform multiple types of analysis on the same data set to demonstrate that your conclusions do not depend on the specific analysis that you've done. And what you can also do is perform meta-analysis. So instead of analyzing data from one singular research group, if you have access to data from multiple published studies, then you can kind of combine them to be much more robust in your inference. Right. So these are the problems, and I'd like to conclude by giving some outlooks on the future of this field. So 3 specific things that I think are going to be fairly important in improving fMRI research and inference are going to be, well, first of all, what I've already alluded to is the importance of connectivity measures and of network neuroscience approaches. So really, neuroimaging is a really useful tool to give us a more holistic perspective on brain function, instead of trying to only find, only localize brain activity to individual parts of the brain. What we can try to begin to unravel is how the individual parts of the brain communicate with each other, and how through this communication they give rise to mental activity. The second important thing is that neuroscience has been entering into the big data era as well, and the use of large-scale databases has been ever increasing, and this of course is a massive opportunity for much more robust statistical inference to be done. And finally, one approach that we'll again see in the reinforcement lecture, hopefully, that I'll give in a couple of weeks, are so-called model-based analyses of fMRI data. So what this means is that in the standard way of doing fMRI experiments, you just have a behavioral task that you ask people to engage in, like perspective taking, if you're a dead fish, and you contrast various different conditions of the task and see whether the bold signal is increased in particular regions of the brain in different parts of the task. But what you can also do is specify a computational model of the mental process that you think the person is engaged in, and you might fit the computational model to the behavioral data, and then use fMRI to maybe locate certain variables, so certain computational variables in your model in the brain, which gives you a whole other kind of perspective, and a whole maybe a more mechanistic approach to trying to identify what kind of mental operations people are actually doing. And I have reached the end, really, of the presentation. So yeah. So thanks everyone for sticking to this. In the end, I also have a couple of slides on a particular study that I wanted to highlight as being, I think, a good example of recent fMRI research. But I'm not going to go through this because it's already 9 o'clock. But you'll have access to the slides, and you can go through them as well. It's this article by Zulham and colleagues, which investigates the neural effects of placebo analgesia. So when people are given only verbal instructions as a form of pain relief, and there's no actual procedure done, and they wanted to see whether this actually did decrease the response to pain in various brain regions, and they showed that it did, and moreover that this correlated with self-reported pain across 600 people, because this is a meta-analysis, so it's fairly robust. And I just wanted to highlight this study as being an example of what I think is a great current study that uses this method. So you can find much more on this and the link in the actual slides. Right, so we're at the end here. Does anyone have any questions? Right. Okay, cool. Well, thanks for sticking to this, and hopefully I'll see you in the next lecture, and possibly in the next lecture that I give, which is going to be on reinforcement learning at the end of I think at the end of the month.
Thank you,
Right.
Bence. Thank you.
Thank you very much.
Terrific, your presentation. And I'm gonna stop the recording.
Yep, and